Very nice to be back in St. Louis again. I was here drive by train, and you come over the bridge and you see the arch. Then he was very upset because, of course, it wasn't at all. And my group should be watching at the same time. What I want unusual, spectroscopically unusual LH2 complexes. Before I start, I should people from
because they cannot hear it yet.
I just hold it and speak into that. Is that better? I'll do that, and I'll try to remember to move it when I turn my head so I don't come out. Okay, so what I want to do to begin with is to thank all the people who've been involved in this work, especially my very good friend Neil Isaacs, who's the protein crystallographer in Glasgow, and a large number of people who've worked together with us over many years on this project. Also, Miroslav and Anna, who were at the synchrotron at Daresbury, where a lot of the data for this was collected. Then Jürgen Kohler and his group in Bayreuth, where we've been doing the single molecule spectroscopy. So this is a cartoon of the membrane of these purple bacteria, and they essentially have a cut-down version of photosynthesis. Single type of reaction center, two types of light harvesting complexes, simple cyclic electron transport, pumping protons. The proton gradient is consumed by the ATP synthase, and you make a... I'm old enough to have started working on this system when all of these were just black boxes. Nothing, essentially, apart from the fact that there was cyclic electron transport and there was this. Now we have pretty good X-ray structures for all of these components, and it's been an amazing experience to live through that change and also to be having been involved in some of the experiments that have elucidated these structures. Now, I'm interested basically in understanding the very early reactions in photosynthesis, light harvesting and the transduction in the reaction centers. And I've always worked with purple bacteria rather than plants, mainly because they're simple. Now, you can grow the bacteria all times of the year, which is nice. The purple non-sulfur bacteria can be made photosynthesis minus by a variety of mutants, and you can rescue them by growing aerobically. But most importantly for me, their pigment protein complexes are very robust, so when you make them, it's easy to handle them and to work with them. And also, all of the pigment groups in these pigment protein complexes are well separated spectrally, so if you want to do functional work, you know what you're exciting and you know what you're measuring, as opposed to higher plants where there's terrible spectral congestion in the long wavelength chlorophyll A and B bands, and it's very difficult there to have that certainty in the experiments that you can have in the purple bacteria. Now these bacteria are very flexible, and very many species are able to not only adjust the amount of these complexes that they have in the membrane, but also adjust the type of LH2. For a long time we worked on a rather standard type of LH2 complex, which I'll describe to you. But with certain bacteria, if you grow them under different light conditions, especially low light, they will exchange the type of LH2 complex that they have, and that enables those bacteria to grow at much lower light intensities. Now, very often, these extra types of LH2 are spectrally unusual. That is, they're non-standard compared to the one we first described the structure of. And if you go out in the wild and you look at the variety, there's a very large natural variation in the spectroscopic forms of LH2. And we would like to take advantage of that to understand the structural basis of that variation, and then try to understand what the organism's benefit is from being able to synthesize these other types of LH2. How does that extra type of LH2 enable them to grow at such low light intensities? So that's what I want to talk to you about today. So it's very easy working with these bugs. If you grow them up, break open the cells, get the membranes, all the light harvesting complexes and reaction centers are in these cytoplasmic membranes. You can solubilize them, put them onto a sucrose gradient, turn the centrifuge on, go home, come back in the morning, and as long as you aren't red-green colorblind, then you can find them because they're nicely pigmented. Now, your eye stops seeing about here, so you don't see these long wavelength absorption bands due to bacterial chlorophyll. You see the absorption dominated by the carotenoids, and that's why you see them as orange, brown, purple, and red species, because you can't see. Now, it's very nice with these complexes because if you do something that's harsh to them and denature them, then these absorption bands due to the carotenoids and these due to the long wavelength bacterial chlorophyll absorption bands shift to the blue, because when they're non-covalently bound into these pigment protein complexes, all of these absorption bands move to the red. And your eye turns out to be exquisitely sensitive in this region of the spectrum here where the carotenoids are. And a few nanometers change, which may look insignificant in a spectrophotometer, produces quite a different color by eye. So a trained eye at the bottom of the column is very able to determine whether these are in good shape or they're not. Now, monomeric bacterial chlorophyll A, free in solution, absorbs in the long wavelength absorption band at about 770. In these complexes, you can see in LH2, it absorbs at 800 and at 850, in LH1 at 875. The little peaks here come from the reaction center. This differentiation of where that absorption band is reflects the different organization of the pigments in the protein, from pigment-protein interactions and pigment-pigment interactions. And those interactions are very important because you know the blue end is more energetic than the red end, and this sets up an energy gradient in the system. So if you excite at 800, energy transfer goes downhill to 850, and then downhill from 850 to 875, and then into the red. This sets up like an energetic funnel that targets the energy going downhill spontaneously towards the reaction center. Functionally very important. Now this is a picture from a species called Rhodopseudomonas acidophila, and if you grow them at low light, then they will change the type of LH2 complex to produce an 800, 820 complex. Now, this change occurs because this organism contains multiple gene pairs for the alpha and beta apoproteins which form these complexes. And what happens when you grow them at low light is they turn off the genes for the high light complex and they turn on the genes for the low light complex. So they switch. Now, we've been able to study this complex and indeed determine its structure, and I'll describe to you what the origin of that spectroscopic shift is. Now, I've got to take a step backwards in time now and describe to you the structure of these complexes, and then describe to you the variation that's now being found, and what is the possible origin of those variations. So this was the crystal structure of the LH2 complex, the highlight complex from Pseudomonas acidophila, and you're looking down on the top of it now as if you're looking down on the top of the membrane. There's no hole in the middle, of course that's filled by phospholipids in the membrane, and it's an alpha-beta nonomer. There are 9 alpha-beta pairs, and these very short hydrophobic apoproteins, normally somewhere between 50 and 60 amino acids, are just able to cross the membrane once as a single membrane transpanning alpha helix, and they form like an extended cylinder within the walls of which all the pigments are organized to produce the structure that we know of the light harvesting complex. Now, quite interesting to go back in time, to take yourself back to 1983. Some of you may not have been born then, but I was on sabbatical in Munich, and happened to discover that there was someone called Hartmut Mickel who discovered how to crystallize membrane proteins. I persuaded him to allow me to try to crystallize a whole range of these LH2 complexes, and this one we got crystals from. Now that was in 1983, and I didn't realize how difficult it was going to be to actually determine the structure. Bobstead finally came out in 1995. All the way through that process, Hartmut Mickel had also crystallized another light harvesting complex from a. Its structure turns out to be very, very similar. If you look at it quickly, you won't see a difference, but it's an 8-mer, not a 9-mer. And we had an arrangement that whoever got the structure first would give the other one the coordinates to allow them to solve that structure. And you can imagine what it was like being a relatively young scientist trying to compete with someone who's won the Nobel Prize. We would meet at meetings, and I would sort of sidle up to him quietly and say, no progress. And as soon as he went away, I would go, yes. And then, luckily, we were able to solve the structure first, and I had the great pleasure of sending him the coordinates to him and Klaus Schultem to solve this structure. But you can see there's a variation. The ring size can vary, and we don't understand what controls ring size. We don't understand whether there's any significance to change in ring size. Obviously, it will affect the packing of these complexes in the membrane, but we don't understand whether that's important or not. So that's one of the. Now, if you look at possible origins of variation, I've described to you so far 3. One is that you can have rings with different apoproteins. So you can have the highlight complex from Acidophila, which is the 800-850 complex. The low-light complex, which is 800-820, the apoproteins have changed. You have rings with different sizes, Garnum versus Acidophila. These changes here, change in different apoprotein, requires a multi-gene family. Many species have upwards of 4 to 6 copies of the alpha and beta apoprotein genes for LH2. And that also raises the possibility that maybe you can have rings with heterogeneous apoprotein composition. Why should they always be homologous? And I'll show you at the end of the talk that we have evidence that this is actually true now, and I'll describe to you the great deal of variation. And once you open your mind to that, then you can see that there is actually the possibility for a great deal of variation. Also show you evidence we've got that there may be really quite different ring sizes out there as well as some of the natural variants. Now, I should at this point say something which is a warning, and that is that these organisms are extremely plastic in their ability to adapt to different growth conditions. And which of the apoprotein genes out of a family are turned on are controlled by at least two very distinct regulatory pathways. There's an all or nothing switch, which is usually oxygen. If there's oxygen there, they grow by respiration, and none of these guys are made. If there's no oxygen, they turn on all these genes for photosynthesis. Then the amount of synthesis is controlled by the light intensity. And also it's been found recently that many of these organisms contain a whole array of bacteria phytochromes. So it's a very complicated system of regulation. And if you're collaborating with someone who's providing you with samples, you need to check very carefully how they grow their bacteria, because you'll get very different preparations depending on the way in which you grow the bacteria. And as far as I know, no two labs grow their bugs the same way. And so you're liable to get quite different complexes from apparently the same species. And it's worse than that as well, because we all grow these bacteria in 24-hour light. Out there, they grow in a night-day cycle. And if you grow them under these 24-hour light conditions, they will subtly change their phenotype as you grow over and over again. And so, again, you may think you're working with the same species, but actually you may find a complex with different apoproteins. And if you're doing very high physics and can detect these changes, yeah, there's problems in the literature because of that. So what I'm going to do is take you through the structure of the LH2 complex, and then we'll go on from there. So this is looking sideways now, as if you were in the plane of the membrane, and you can see the transmembrane helices, and you can see the different groups of pigments. These are the chlorophylls absorbing at 800, these are the 850 ones, and the brown ones here are the carotenoids. What I'm going to do first of all is to walk through the structure from this way to describe the organisation of the pigments. Then I'm going to show you a movie to build up the structure, as you can do in a computer, and see how it's constructed. So the first group of pigments you come across are a group of monomeric chlorophyll molecules. These are the 800 absorbing chlorophylls. And the central magnesium atom here is complex to an extension of the N-terminal methionine from the alpha apoprotein. Now, when we first described the structure at a resolution of 2.5 angstroms, we thought this was a 4-mile extension, but we've now got better data by cryocooling the crystals to just about 2 angstroms resolution. And you can see the electron density here is bifurcating. It's not a 4-mile group. We think it's a CO2 adduct on the N-terminus there. It's a rather unusual modification of the protein, but you find that both in xanthine oxidase and in hemoglobin. These monomeric bacterial chlorophyll molecules are the 800 absorbing ones, and if we come out in resolution, you can see that there are 9 of these, 1 for each alpha-beta pair, and the bacteriochlorine rings lie flat within the plane of the membrane, and they're centre to centre separated by about 21 angstroms, and they behave largely as monomeric chlorophyll molecules. Now we know that's true because biochemically we can take these guys out and you lose the 800 absorption, and you can add back those chlorophylls again and you get back the 800 chlorophylls. Now that's important because when you determine the crystal structure and you see pigments in there, they don't have a little flag there saying I absorb at 800. You actually have to do some work to show which group of chlorophylls absorb at which wavelength. And these ones have all the characteristics of being monomeric, and that extraction reconstitution proves it very nicely. When you go further into the structure, roughly in the middle of the structure, then you can see another group of chlorophyll molecules in their electron density. Now these are with their rings parallel to the transmembrane helices, and these are closely interacting, centre to centre, about 8 to 9 angstroms, but almost in van der Waals contact here. These are connected to the protein by histidine residues to the magnesium atoms, one coming from beta, one coming from alpha. So there are 2 of these with each alpha-beta apoprotein. And if you look then, here they are, one complex to alpha, one to beta, and the rings form almost like the blades of a turbine in the structure. These are the strongly interacting 850 chlorophyll molecules, and they essentially are so strongly interacting that they behave like a supermolecule and share the excited states. Right, this is a movie of the pigments associated with 3 of the alpha-beta apoproteins. Here are the 850 chlorophylls, here are the 800 chlorophylls, and here are the phytol chains. If I showed you all of the pigments, you wouldn't be able to see it because there's just too much in this high symmetry. One thing to note is that the way in which these phytol chains interact with each other, this 800 chlorophyll is rather peripherally bound, that's so we can take it out, but it's held in by this very nice interaction between these phytol chains. Now you can see the carotenoid molecule here. Now this carotenoid molecule is very important because it starts its life in one alpha-beta apoprotein, crosses over into the next alpha-beta apoprotein, and sort of bolts the alpha helices together. And Neil Hunter, who should be in Sheffield as we speak now, did some very nice work showing that if you delete the genes for carotenoid biosynthesis, then the LH2 complex is not stably assembled. You find that you can make the complex, but it degrades. It requires this carotenoid to be in there to structurally hold the thing together. And the carotenoid has 2 functions: an essential function, which is to protect the complex from photodynamic damage. If you illuminate chlorophylls in the presence of oxygen, triplet chlorophyll will sensitize singlet oxygen, and that's very dangerous. But because these carotenoids are in van der Waals contact with the 800s and the 850s, any triplet states there are rapidly quenched by the carotenoid. They beat the oxygen there by 3 orders of magnitude in time and give a great measure of photoprotection. They also can absorb light at the singlet level and transfer it to the chlorophylls, making these bugs able to use green light as well as the far red light to sensitize their photosynthesis. This carotenoid here is one called rhodopene glucoside. It's unusual, it has a sugar residue. It's in the all trans configuration and actually has 11 conjugated double bonds. Now this is an interesting artifact that came with the higher resolution structure. It looked like that there was a second carotenoid molecule here in a terrible twisted cis conformation. When you determine the structure of a protein in a crystal, you assume that all the protein molecules within the crystal are identical. And what happened in this case is some of the proteins had LDAO, a detergent molecule bound, and some of them had octoglucoside, another detergent bound. And when you average overall, those 2 come together and it looks like you've got a second carotenoid molecule. It's not. It's 2 detergent molecules there, and that was shown by Miroslav, who crystallized again using cubic lipidic phases and was able to show that these came from detergent molecules, not from a second carotenoid. So here you see now the alpha helices holding the whole structure together, and the alpha and beta apoproteins only touch on either side of the membranes. They don't come into contact at all in the actual plane of the membrane, and it's a very oily structure. It's held together as much by pigment-protein interactions as it is by protein-protein interactions. It's a very If you're interested in how these things work, you've got to look at their orientation and organization of the chlorophylls. Now, although the chlorophyll rings from 800 and 850 are orthogonal to each other, the long wavelength transition dipole moments are actually rather parallel to each other. So that's very good for singlet-singlet energy transfer. And if you excite one of these chlorophyll molecules here at 800 with a short flash of light, let's say 100 femtoseconds, it takes 0.9 picoseconds for the energy to drop down into the 850 band. And If you cool these complexes down to liquid helium temperatures, that only slows down to about 2 picoseconds. Essentially, this is a hard-wired system. There's no molecular motion required. The pigments are organized, and then energy transfer occurs amazingly quickly and efficiently. It's almost independent of temperature. It's an amazing system. Now, the 800-820 complex had been studied before we got the structure in 2 ways. People had looked at the sequence and detected that 2 residues which could potentially be hydrogen bonding residues, a tyrosine and a tryptophan, to the chlorophylls, were replaced by phenylalanine and leucine. And resonance Raman work in Bruno Robert's lab in Paris had shown that when you compared these two, these guys had the phenine acetyl carbonyl group hydrogen bonded, where these ones didn't. And Neil Hunter and his group in Sheffield were able to use cycle-directed mutagenesis and to make those changes and convert the Rhodobacter 800-850 complex into an 800-820 complex. We were able to crystallize this complex, and it's very similar. You can almost completely overlay it with the 800-850 complex. But if you look in detail here, let's just look in this one case here. This acetyl group, which in the 850 complex is within the plane, is now in the mutant out of the plane. And what's happened is that acetyl group has rotated out of the plane of conjugation. And it's like the old particle in a box. You take out one extra conjugated double bond, the site energy moves to the blue, and the complex now absorbs at 820 rather than 850. It's almost a very subtle change in the structure, allows you to twist the angle of that carbonyl group and produce that site energy change to produce the 800-820 complex. Jacques Fager had worked on this many years ago on artificial or special synthesized porphyrin type molecules and shown that this mechanism actually works in a more general way. And here was the bacteria taking account of that mechanism. Now, recently we've been working on a species called Rhodopseudomonas palustris, and when it's grown at low light intensity, it has this strange complex, which is 800, 850, but now the 850 band is very small compared to the classical acidophila type complex. And we've been interested in trying to understand what that might be due to. And I should say something about this complex before, because Miroslav and his group in Daresbury had produced a low-resolution crystal structure for this complex. Strangely enough, it didn't have the 850 band here, just the 800 band, and they suggested this would be an octamer and also contain an extra chlorophyll molecule per alpha-beta pair. So we had that as a background before we started, and we wanted to look at this. But that's not the only thing you can get, because if you go to cremation venosum, then you can get even more variance spectroscopically. So here's a sort of classical looking highlight complex, but actually the 800 band has 2 peaks in it, not just one. You can have an 800-840 complex, which is like this one. You can have an 800-820 complex, and an 800-850 where the 800 band is more intense than the 850. Extremely complicated. And it's very difficult to isolate and to separate these different complexes. And they tend to be made simultaneously, and so it's really difficult to work on this system. Now, one thing that we've noticed, if you do molecular sieve chromatography, all of these are done under identical conditions with DDM as the detergent. Then you can see the acidophila complex, which we know is a monomer. Here's the 800-850 cremation venosum complex, which is probably a monomer as well. It's not completely separated from the 800-820, but the 800-820 is clearly a different molecular weight form. The earlier you come off the column, the larger you are, and this suggests this ring size is bigger than a 9-mer. And Andrew Gould, working in Bruno Robert's lab, has suggested that this might even be as big as a 12mer. And we're very interested to actually see if we can work out what the size of that ring might be. So there's a lady called Carol Robinson in Oxford University, has a very nice mass spec system for allowing holo membrane proteins to run in the spectrophotometer, have the detergent micelles stripped off them, and then calculate the real size of those complexes. So we've sent these complexes to her, and we're waiting to see whether she can tell us what the ring size of this is compared to the ring size of these guys. Again, I should emphasize, we don't know how the ring size is controlled, and we don't know whether that's important functionally for the system. But obviously there is the possibility of getting different ring sizes. Now, there's a question. If you're going to want to work on these complicated systems, and it's not really possible to separate these different complexes, you have two possible situations. You can have mixtures of complexes that you can't separate, or you can have complexes which are heterogeneous. So some of the complexes have mixtures of different types of apoproteins. So if you can't separate them, how can you distinguish between these two situations? That's a question for you. How can you distinguish between the two situations if you can't separate them? You've got these two possibilities, a mixture or heterogeneous. Come on, Aaron. This is a test for your PhD. That's one way, but we're not clever enough to do that. There's a simple way to do it. How would we do it? That's the answer. Well done. Yes, to investigate by single molecule spectroscopy. So this is the driver for working with Jürgen Kohler in Bayreuth. He's very good at doing single molecule spectroscopy. And if you can look at the properties of individual molecules, then can you get evidence for heterogeneity in a single molecule, or do you see 2 or 3 different classes of single molecules from a mixture? So that's what we set out to do. So what you do is you dilute the complexes to very high dilution, you spin coat them down onto a surface, freeze them down to liquid helium temperatures, and then under a confocal microscope look and see the different individual LH2 complexes emitting light. Now you select an individual complex, which you can do because all the individual ones appear the same size and have the same intensity of illumination, and you can measure their emission spectrum. And then, if you want to get more information, you can determine their excitation spectrum, measuring the emission in the red end. So you scan through, measure the emission here, and you can measure the excitation spectrum, which is equivalent to measuring their absorption spectrum. Now the first thing you have to do is do some controls, because if you're looking at a single molecule, you don't know it's a good one or it's a dead one. So you have to accumulate enough single molecules and add up the spectrum to get back to the ensemble spectrum. So at low temperature, here is the ensemble spectrum of the highlight LH2 complex from Acidophila, the dotted line. And the jagged line is adding up about 50 individual molecules. Now in the ensemble, all the bands are broad. But in the single molecule spectrum, you see remarkably in the 800 region, sharp individual bands and broad bands, either 2 or 3 in the 850 region. Now, these come from those individual monomeric bacteriochlorophyll molecules. They have potentially a long lifetime, therefore they have narrow bands in the single molecule spectrum. These broad bands reflect the fact that this absorption band reflects exciton coupled states, and so you get broad bands. Now, I can show you a nice movie of this. And if you look here in the 800 region, as you change the polarization of the exciting light, the different 800 chlorophylls appear and disappear in the spectrum. Because if you think about them being on the surface, they have different organization, and as you change the polarization, you interrogate different chlorophyll molecules around the ring, and you see that. And here you see the organization of those broadbands. So if you actually look at the exciton bands that are possible with this 9-mer symmetry, you find that most of the oscillator strength lays in these two bands here, the K plus or minus 1 states, and theory predicts that these two bands should be orthogonal with regard to polarization to each other. So these two spectra here are taken with orthogonal polarization, and you can see these two bands very nicely in the single molecule spectrum. And so it's a very nice proof of that type of description of what's going on. So what happens when we go to these clusterous complexes? Well, the clusterous complex have a horrible, large, multi-gene family for both the alpha the apoproteins and the beta apoproteins. And all of the complexes, as we prepare them, have many of these gene products present. So what happens in the single molecule spectroscopy? Again, the ensemble spectrum and the one by adding these guys up. If you go in the 800 region, then you see nice sharp bands. In the 850 region, you see the broad bands, but you also now see broad bands in the 820 region. So how can you have this extra complement of exciton coupled bands in this sort of structure? And what's the origin of that? In order to understand that, you have to do some simulations and molecular modeling for this. And my student Tatus, who should be in Glasgow listening to this, did lots of these simulations. And what he did was to try to simulate, first of all, the spectra. And you do the simulations by having different ring sizes, by different degrees of disorder. And he found he could fit the spectra very nicely with either an 8-mer or a 9-mer ring structure. Now, remind you, we were expecting 8-mer to be the right answer. But if you get more information from these, which you can get by recording spectra and changing the polarization, you can get the different bands in the spectrum, and you can see their angular dependence with regard to the polarization of the exciting laser. And you can see three bands, B1, B2, and B3. And what he did was to collect information on the energy gap between B1, B2, B2, B3, and the angular difference in their polarization properties, and produce a large amount of information in these charts in terms of the energy gaps and the angular difference. And now try to simulate again and see what type of simulation can match the data. Now, I should emphasize that when you do these simulations, you can't prove a model is true, but you can disprove something if it doesn't fit. Now, what he tried was to use 8-mers, 9-mers, 10-mers, mixtures of 8-mers and 9-mers, mixtures of 8-mers, 9-mers and 10-mers, and all the combinations you could think of. And also, the only way that he could get good data was, or good simulation to fit the data, was by assuming that some of the site energies in the ring were 800, 820, and some were 800, 850. And the best fit that he could get was this model, where there were mixtures of 800, 820 and 800, 850 in the ring like that, and it was a 9-mer. Now, the fits are not perfect, so this is probably close to a real situation. We've no idea whether this type of order in that structure is correct. I can't see how that would be formed biologically, but it fits the data. But there may be variants on that organization that cause this fit not to be very perfect, and we may have to play with mixtures of these fits to get that. But the thing that you can guarantee from this is that it's probably a non-numeric structure, and the only way you can get anywhere near a reasonable fit is by assuming the rings are heterogeneous. That some of them have 800, 820 character as polypeptides, and some of them have this 800, 850 character of polypeptides. So this extra degree of flexibility is there in these structures, and it's beautifully confirmed by this single molecule spectroscopy. So we have quite a nice picture now of what's going on in the membrane, and if you've read any of the papers coming from Neil Hunter's lab or from Simon Schuring, they've got these beautiful AFM pictures of these purple bacterial membranes where you can actually see the LH1 and reaction center and the LH2 complexes within the membrane. And so we have a great deal of knowledge now in terms of all of these potential energy transfer reactions having been time resolved, and you can see how you can vary the ratio of LH2 to LH1 and the type of LH2 by how you grow the species, which species you're working in. And what we'd love to be able to do, and this is a very difficult thing to try to do, is to map the organization of these guys in a very limited surface area of a single membrane, and then to be able to time resolve the energy transfer reactions in that area where you know what the structure is, and then go to the next piece of membrane where the structure is quite variable and different, and then come and do the same thing. One of the nice things about these complexes is because they're highly symmetric, it's just as easy to take energy out of here or there or there or there. And so there's a great deal of flexibility potentially in how you would organize these complexes and still have very efficient light harvesting. And if you look at the membranes, especially some of the ones that Neil Hunter's described, they're very heterogeneous, and yet they still work apparently very well. And I think it's because of this ability that as long as the rings are close enough to each other, there doesn't have to be high order for the system to work very well. And what I'd like to do is to leave you with a video that Tatas made showing the energy transfer reactions. Now, it's a very nice little thing to look at. It's probably wrong in terms of the fact that this excited state shouldn't just be whizzing around, but they're delocalized. But it gives you the idea of what happens when you're able to excite into an LH2 and then get ring-to-ring transfer and then energy transfer coming in there, and then the active branch of the finish with that. I think it's fair to say that we know.
You will have to repeat that.
Okay, right. The question is, do we know anything about the assembly of these complexes? Do you need to have the pigments there first, or do the alpha helices come in and then the pigments? I think the answer is we know nothing about the assembly. One thing that's clear, if you look at membranes when cells are growing, you don't see bits and pieces. It looks like it's a highly cooperative effect. But how that's controlled, I don't know. Anybody else does know either. Okay, so the question is, what do the organisms gain by having these extra forms under low light? Again, I think the honest answer is that we aren't sure. The one thing you can demonstrate is that those bugs that do this will grow at significantly lower light intensity than those that don't. That's switching the type of LH2. If you measure the forward energy transfer rate from, let's say, the 820 into the LH1, it's marginally slower than the 850. But if you measure the back transfer rate from LH1 to 820, it's much, much slower than the back transfer from 800 to LH1 to 850. So it appears that the advantage is somehow being able to confine the excited state to the LH1 ring for longer, I guess so that if a trap was shut, it can wait until the trap opens. And so you trap more effectively all of the photons that come in. Whether that's true or not, we'll have to wait for the experiments. Do the have a similar quantum yield? I can answer that question as we haven't actually measured quantum yields because that's quite a difficult absolute measurement to make. But what you can show is the excited state lifetime of either the 800, 820 or the 850. And those in both types of complexes are on the order of a nanosecond. So it doesn't seem to be a significant lengthening of that excited state lifetime. We haven't measured quantum yields. Stop me when you get to the one that... no, I'm not clever enough to do that. It's okay.
You got a lot of them, so keep going.
That one, okay. The first model.
I assume that if that were for Brodus Barone,
but no LH2. Rubrum is one of the species only has LH1 in reaction centers.
so the question is, in the early
Right, here we go. There's a question which we might call a blast from the past. Is in the early 60s, in rubrum, under illumination conditions, you could see free radicals. Okay. I suspect that what you were seeing to begin with was the G=2 signal from P+ in the reaction centers. Because when you illuminate those membranes, you would oxidize the chlorophyll. And if you had sufficient intensity, you would maintain the special pair oxidized. So I suspect the predominant signal you would have seen would have been the special pair cation from P870 in the reaction center. You can do other things that damage the system and produce other free radicals, but that's probably the dominant one you would have seen. It would have been reversible in the light.
I'm sorry?
Was it?
Yes, oh yes.
So then that was from P+.
Yeah, well, we were close, except that we had no.
Right. The longest wavelength absorption of bugs that I know about comes from species that contain bacteriochlorophyll B. And although bacteriochlorophyll B in organic solution is a monomeric species, only absorbs at about 20 nanometers beyond that of bacteriochlorophyll A, 790 versus 770. In the membrane, in species like Rhodopseudomonas viridis, the LH1 absorption band is out at 1,012 nanometers. That's the longest one that I know. There are species containing bacteriochlorophyll A that have absorption bands that go out to about 980. But they're extremely difficult to grow, and no one's really worked on those significantly. So it's bacteriochlorophyll B. Himadri.
So the
So this is a question of what you would see in the single molecule spectra when you have the homogeneous case. This is what you see. Okay, so these are the spectra from LH2, the highlight form from acidophila. Now, you see a lot of individual variation because every time you freeze down an individual complex, you can freeze it in a slightly different state, and therefore you can see differences in the spectra. Okay? But when we analyze all of this data very carefully, you can model and completely explain the data on a homogeneous case. And the key is that you don't have these extra bands in the 820 region. All of those bands you could explain from distortions of the homogeneous case, 800, 850. No, what I said was that the retention of the excited state in LH1 in membranes is such that you don't lose it back out to the pool of LH2 again.
Right, so
these bugs grow anaerobically. So all the work we do is in the absence of oxygen. So actually carotenoids are not essential under those conditions. They're essential to form LH2, but if you make a carotenoid minus bug, typically they only have LH1 in the reaction centers. So the LH1 is stable in the absence of the carotenoid. The reaction center is stable in the absence of the carotenoid. Introduce oxygen and light, they're dead. So it's not, that answers the question.